is your data set downloaded now? Is it bold? Yes. Okay. I want you to click it. Double click. Is that the one? No? Where did I put it? Oh, did I make a new project? Oh, I put it in the intro to bio. Oh, well. Okay, double click it. All right. Oh, wait. It's coming. Has everybody's come up yet? Everybody see this window here? Yeah? Okay. This is what, and this is kind of the summation of all that stuff. So use your comparison analysis once you've downloaded everything in. Go through here, look at this comparison analysis and figure out what might be interesting. Canonical pathways. Protein kinase A signaling. Google that in lung cancer, see what happens. Glycolysis. Let's take a look at glycolysis for a minute. And you all remember glycolysis, right? Yes, I love talking to high school kids because it's fresh in your mind. You talk to grad students, they're like, sorta. Okay, open pathway. Remember, if it's red, it's going up, or it's increasing with as the cancer gets worse. Look at all these enzymes. What color are they? Pink, red, yeah? So that means they're up. What is this? If you were, try to put this in the context of cancer biology. What is this telling you about these tumors? All these enzymes that are converting glucose phosphate into components of the Krebs cycle. What is that telling you about these cells? What are they doing? Say that again. They're eating. They're eating a lot. This is a teenager that's like hitting a growth spurt and going crazy. This is basically them eating potatoes and Pop-Tarts, and they're getting their sugar. We can see that at this level. Just based on, remember, we took a blind knife, cut it, and now we can tell that these tumors are utilizing more glucose. And it makes sense, right? And potentially, if we wanted to use this as a therapy, maybe we target one of these enzymes. Although your other cells need energy too, so that might be difficult. But you can kind of see what I'm talking about here is, what are the pathways that these things are utilizing? Now let's find its weakness. How does it work? Once I know how it works, then I can throw in that wrench to mess up the system. Let's see some of these other upstream analyses. P53 is the top one. Out of those 900 and some genes that were in at least 2 of those studies, it interacts with 126 of them. And again, remember this column as you're going through here. Okay, pay attention to this. Mechanistic network on this side. Basically what they're doing is they're taking all these godfathers and they're putting them into like the Gambino crime family kind of diagram. Check this out. What you're going to see is basically the skeleton that connects all this language together. P53 is supposed to activate this one. It's not working. It's going down. This tumor suppressor is going down. It's supposed to inhibit ERB2, which promotes cell growth. It's going up, which is activating all of these things. This one actually condenses chromosome. It's getting deactivated so that these cells can divide. And you can look down here. Here's some other tumor suppressors as well. Here are some important transcription factors that this lung cancer is using. Again, this is supplying me, this is kind of like the crib notes, or you could think of it as the scout that goes out, ahead of the expedition. Like I'm saying, hey, this is where, there's a path here, there's a mountain here. I'm basically giving the scientists I work with kind of a heads up of what's going on, and now they can test this stuff. And a lot of times when I show them graphs like this, they're like, the first thing they do is they go, oh my gosh, I never knew this thing was up. That's crazy. Okay? So here's what I want you to do. By tomorrow, I want you to find a gene based on all those lists that I gave. I want you to find a gene and be able to talk about it and how it relates to lung cancer. Okay? And I'm thinking everybody will probably pick a different gene. But I want you to go there and show me the research, why you picked this gene. How does it relate to lung cancer? Okay? All right, so for the next bit of class, why don't you start downloading those studies in there? And we will meet, what do we say? Wednesday at 2 p.m. at UC Denver. Okay? Well, the campus.
Oh, I'm about to say, I'm like, all the way over there?
Yeah, over there. And bring your big car so you